So amyloids are protein, very tough aggregations of proteins that are fibrous in nature. And it turns out that these prions that we've been working on, when they pile up like this to form the prion fiber, it's actually forms a protein structure that's very, very related to the kinds of protein structures that we actually see accumulating in the brains of people who get these terrible neurodegenerative diseases like Alzheimer's disease. A beta is, you've probably heard of A beta plaques. Plaques are made up of these A beta underlying fibers of this peptide called A beta. And the way in which they're structured is very similar to the way in which our proteins, these prion proteins, acquire this structure. So that's one of the reasons why we actually first decided we would start to attack these neurodegenerative diseases, because we knew that the nature of the protein folding problem was something that we could use a simple yeast cell to look at and discover. So we've actually taken some of these proteins that misfold in the human brain, put them into our yeast cells. They misfold in the yeast cells. They create problems of exactly the same type that they do in our brains. And so we can then go and try to find things that will stop them from misfolding, stop them from interacting with other things in the cell, and try to control the terrors that they unleash. One of the reasons why amyloids are so difficult to study is that they can't be looked at in the ways that protein structure people have devised for normal proteins. So we have X-ray crystallography, we have nuclear magnetic resonance, we have different ways of looking and seeing the structure of proteins. And those were all developed and devised with proteins that behave themselves and stay nice and solid and stay individual and will line up like this in a nice orderly way if necessary. But those methods don't work for these amyloids. So we have to devise and create new methods or push those existing methods to the absolute limits. And one of the things we've been trying to do then is to use genetic methods to kind of decipher what's going wrong with those proteins, why they're folding. We change one part of the protein and see if that can change, and that can help us understand something about the structure. And we use a variety of other biophysical techniques in my lab as well that are based on trying to get at what these things are doing, how exactly they're misfolding. And it's really because with the normal ways in which we have of discovering structural insights don't work on them. And there's one other reason why it's really tough, and that is that the really bad stuff is probably not the stuff that's piling up in these nice orderly arrays. It's these early misfolded things that are then prone to interacting with other stuff. So they start to misfold and they start interacting with a bunch of other stuff, and they're dynamic. The reason they're so bad is that they're dynamic. They're interacting with lots of other stuff, but the fact that they're so dynamic makes them awfully hard to get a visual picture of. So there's really two problems that are involved. The thought that these amyloids might be just clogging things up. And I actually even used to use that kind of analogy because it's an easy one for people to see. But it's probably not that they're clogging things up as much as that they're causing all sorts of reactions to occur that shouldn't be happening. All sorts of things are, one protein is interacting with another protein and it shouldn't be doing that. And it's that kind of chaos that misfolding can cause that's really at the heart of this, and it's causing neurons to do things that they shouldn't be doing.